Chapter 13 Photosynthesis in Higher Plants. Summary. Green plants make their own food by photosynthesis. During this process, carbon dioxide from the atmosphere is taken in by leaves through stomata and used for making carbohydrates, principally glucose and starch. Photosynthesis takes place only in the green parts of the plants, mainly the leaves. Within the leaves, the mesophyll cells have a large number of chloroplasts that are responsible for CO2 fixation. Within the chloroplast, the membranes are sized for the light reaction, while the chemosynthetic pathway occurs in the stroma. Photosynthesis has two stages: the light reaction and the carbon fixing reactions. In the light reactions, the light energy is absorbed by the pigments present in the antenna and funneled to special chlorophyll A molecules called reaction center chlorophylls. There are two photosystems, PS1 and PS2. PS1 has a 700 nanometer absorbing chlorophyll A P700 molecule at its reaction center, while PS2 has a P680 reaction center that absorbs red light at 680 nanometer. After absorbing light, electrons are excited and transferred through PS2 and PS1 and finally to NAD, forming NADH. During this process, a proton gradient is created across the membrane of the thylakoid. The breakdown of the proton gradient due to movement through the F0 part of the ATPase enzyme releases enough energy for synthesis of ATP. Splitting of water molecule is associated with PS2, resulting in the release of oxygen, protons, and transfer of electrons to PS2. In the carbon fixation cycle, CO2 is added by the enzyme Rubisco to a 5-carbon compound RUBP that is converted to 2 molecules of 3-carbon PGA. This is then converted to sugar by the Calvin cycle, and then the RUBP is regenerated. During this process, ATP and NADPH synthesized in the light reaction are utilized. Rubisco also catalyzes a wasteful oxygenation reaction in C3 plants photorespiration. Some tropical plants show a special type of photosynthesis called C4 pathway. In these plants, the first product of CO2 fixation that takes place in the mesophyll is a 4-carbon compound. In the bundle sheath cells, the Calvin pathway is carried out for the synthesis of carbohydrates.